Just a brief video lecture on the different levels of protein structure. We have primary structure, secondary structure, tertiary structure, and quaternary structure. So first let's talk about primary structure. This is the easiest level or easiest organization of protein structure that you can envision, because all it is just a linear sequence of letters. It is just a linear sequence of amino acids. So you can look up the primary amino acid sequence of any protein in the protein data bank or PubMed. But it's just a linear chain of amino acids. So let's just say A for alanine, A, Y, D, G, N, S. I hope you know what the I hope you know what the one-letter amino acid abbreviation means for each of these. Otherwise, it's going to be very difficult in future courses, just in future careers. When you go to medical school or PA school, I'm sure this is important stuff to know. So like that, you can go on and on and on with all the different one-letter amino acid abbreviations. So that is just a hypothetical primary amino acid sequence. It's just a linear chain of amino acids. Again, we typically go from N-terminus to C-terminus. So the alpha amino group of alanine starts this protein chain, a polypeptide, and it ends with the alpha carboxylic acid group on glycine. So like that, this could be 100 amino acids, it could be 75 amino acids. I think the largest protein is titin, which is a muscle protein. It's about 1,000 amino acids, so there would be 1,000 of these letters. So that's the primary structure, a linear sequence of the one-letter amino acid abbreviation. It could also be 3 letters abbreviation, but the linear amino acid sequence is the primary structure. Notice there's nothing three-dimensional about it. It's just kind of like data. Okay, it's kind of like some sort of binary code or some sort of program code. It's just letters, but each letter, remember, signifies a specific amino acid. Okay, nothing three-dimensional about the primary structure. We start going into 3 dimensions when we talk about secondary structure. And students have a somewhat of a difficult time understanding where secondary structure emanates from. Secondary structure emanates from hydrogen bonding within the peptide bond. Okay? So when we have that restraint, hydrogen bonding within the peptide bond and the peptide bond alone, I will expand this and say hydrogen bonding within the peptide bond of the polypeptide chain when you allow for those parameters of hydrogen bonding only from peptide bond to other peptide bonds somewhere in the primary amino acid sequence, okay, there's really 2 structures that can emanate. So here's a hypothetical hydrogen bond between an NH of one peptide bond somewhere in the primary amino acid sequence, hydrogen bonding with the nitrogen of a peptide bond somewhere else in the primary amino acid sequence. Okay. So when you have that constraint, where you only have hydrogen bonding within these planar peptide bonds across the primary amino acid sequence, there's really 2 forms that can arise. Okay, so Linus Pauling was instrumental in this. Again, notice the constraint. Okay, it's hydrogen bonding within the peptide bond across the polypeptide chain. So here's a peptide bond somewhere in this amino acid sequence, and it forms a hydrogen bond with somewhere else, with another region across the amino acid sequence. So with that constraint of hydrogen bonding only within the peptide bond or within the peptide backbone, okay, remember, this whole thing would have a polypeptide backbone and then side chains coming out in three-dimensional space. So with that constraint, we actually have two types of secondary structures. We have the alpha helix, okay, and out of the alpha helix, we can have an I+4 alpha helix. We can have an I+3 helix, also known as the 310 helix. And then we can have an I+5 alpha helix, which is known as a pi helix. Okay, so you should know what the I and I+4 mean. Basically, it is a hydrogen bond with a C double bond O, hydrogen bonded to a NH, 4 amino acids away. It's actually more 3.6 residues away, so it's not exactly a full amino acid in which the hydrogen bond occurs. It's about 3.6 amino acids, but we round up and we call it an I+4 alpha helix. So again, hydrogen bonding with the components of the peptide bond, the components of the C double bond O NH, which comprises your peptide bond that links all these amino acids in the primary amino acid sequence, links them together, the peptide bond. So you can have it between the C double bond O and the NH, about 4 amino acids away. That's the standard I+4 helix. You can have it about 3 amino acids away. That's called the 310 helix. That 10 as the subscript implies there's 10 atoms between the hydrogen bond of the C double bond O and the NH. So there's 10 atoms that span that hydrogen bond. And then you have what's called a pi helix, where you have a hydrogen bond with the C double bond O, and then with an NH group of another peptide bond about 5 amino acids away. So I'm not going to go into the technicalities of the alpha helices, but generally speaking, and you can look at the book and consult the lecture, but the alpha helix is going to look like your helix, okay? Here's your polypeptide backbone, and then your side chains would sort of protrude out like this. They can protrude towards you. They could protrude in the back of this piece of paper. Okay, the side chains are also known as the R groups. Okay. We also talked about amphipathic alpha helices. If we haven't, amphipathic alpha helices are very common in a lot of natural products chemistry. That is, if you have your eyeball looking down of the helix, you can actually see what's called a helical wheel, and all the R groups of polar amino acids are on one side, and all the R groups of nonpolar amino acids are preferentially localized on another side. So this is looking down at a helix. That's called an amphipathic alpha helix. And a lot of antimicrobial peptides, also known as AMPs, amphipathic alpha helices. A lot of amphipathic alpha helices have this configuration where the R groups are charged or all on one side, and maybe nonpolar or hydrophobic amino acid side chains are all on the other side. So AMPs are common. A lot of natural products, chemistries like fungi and yeast, bacteria may produce amphipathic alpha helices. All right, so this is called a helical wheel plot, or a helical wheel diagram. I would consult the lecture because I'm not going to go into helical wheel plots in any more detail for this particular video. All right, so we have, under secondary structure, we have an alpha helix, and then we also have the beta sheet. So this is meant to be a very, very quick cursory review. A beta sheet can be parallel or anti-parallel. So typically a beta sheet kind of looks flat, okay, and it has your directionality of N to C. So what I've drawn here is a parallel beta sheet. So one of these would be a beta strand. This is beta strand 1, this is beta strand 2. They happen to be parallel because just the directionality, the polarity is N to N, or N to N, and then maybe you'd have an alpha helix here and a random coil here, and then your C terminus ends right here. So so this would be what is called a parallel beta sheet. You can also have an anti-parallel beta sheet, kind of the same way. An anti-parallel beta sheet would have an N to C configuration. Okay, these are flat. Okay, the polypeptide backbone is flat. So I'd have a strand here, and then here's my N terminus, and here's my C terminus. So this strand is going in this direction, this other strand is going in that direction. They're connected by a random coil. That would be an example of an anti-parallel beta sheet. So one way to think of beta sheets, again, it's hydrogen bonding within the peptide backbone, okay, is a flat sheeted structure where the R groups project up and down this flat sheet. Okay, the sheet comprises your peptide backbone, and your R groups are going to protrude up and down, okay, or above and below the plane of this piece of paper. Okay, the R groups of the amino acids, and then this connecting region is usually a random coil. Again, hydrogen bonding across the peptide backbone. The third example of secondary structure is the random coil. Okay, the random coil really just takes its name from the fact that it's just a random. Okay, no real order to it. All right, so we can just draw a random coil like that. Sometimes it's just called a disordered region. I know secondary structure means hydrogen bonding across the peptide backbone, but really that's your alpha helix and your beta sheet, because those are more ordered structures, ordered structural arrangements when these things hydrogen bond amongst one another across the primary amino acid sequence. A random coil is more of a disordered structure. All right, now let's go into tertiary structure. In tertiary structure, we start introducing hydrogen bonding and electrostatic interactions, polar-polar, non-polar. We start introducing these interactions amongst the R groups. Okay, so bonding amongst the R groups. So remember, there's 20 R groups that comprise the 20 amino acids. So what are these bonding amongst the R groups? Well, we can have hydrogen bonding. Okay. All right, so an example of that potentially would be the side chain of a lysine, perhaps with the side chain of a protonated aspartic acid. So here's the side chain of a lysine. So you have to start thinking of these things in terms of what amino acids would like to interact with one another, what amino acids would want to maybe stay away from one another. Okay, so you're going to have to start thinking about that in these terms. Okay, so lysine, maybe the hydrogen bond can be like this. Okay, so this would be the side chain of a lysine. This would be the side chain of an aspartic acid that's protonated, and here would be your hydrogen bond. Okay. So that's just one example. It's an interaction amongst the R groups. Okay. Another example would be ionic interactions. Okay. Using these same examples again, maybe we're at a different pH. We can have an ionic interaction with the side chain of lysine. Okay, that's a positive charge at physiologic pH, and it may interact with the negative charge of, I don't know off the top of my head, maybe the negative charge of tyrosine. Okay, here is tyrosine. Okay, in which that proton of tyrosine has come off. Okay, so this is a positive interaction, okay, between lysine and the deprotonated side chain of tyrosine. All right. Another one would be hydrophobic interactions. All right. Maybe phenylalanine. There's a CH2 here. Can interact through stacking with another phenylalanine someplace else in the protein's primary amino acid sequence. So we have sort of these benzene rings stacking amongst one another. Okay, so you notice I'm talking about side chains and how they want to interact, or in some cases get away from one another. All right, and there's hydrogen bonding, ionic, hydrophobic, van der Waals forces. Polar amongst the side chain, non-polar. All of these are constructive interactions. They could also be destructive interactions. The big one is disulfide bonds, though. That requires a special mention. Okay, disulfide bonds is actually a covalent bond between two cysteines. Okay, so that is a huge tertiary structure determinant. That's going to put a lot of constraints when the protein folds, if the two cysteines decide to form a disulfide bond. Just because two cysteines are there doesn't mean they'll form a disulfide bond, but if they do, when the protein folds into its native three-dimensional shape, and configuration, when they do decide to form a disulfide bond, that kind of is going to cause other side chains and other interactions to sort of adjust and recalibrate itself. So just to remind you what a disulfide bond is, it's the side chain of cysteine interacting with another side chain of cysteine, forming this very strong disulfide bond. Okay, a huge tertiary structure determinant. Okay, a huge and very important tertiary structure determinant. Okay, disulfide bonds. So just some examples of tertiary structure. Again, we're talking about the R group. So secondary structure really is constrained to the peptide bond and hydrogen bonding. Tertiary structures are bonding with the R groups, and they're not confined to hydrogen bonding alone. They could be ionic interactions, hydrophobic interactions, van der Waals interactions, polar interactions among side chains, non-polar interactions among side chains. The big one is obviously disulfide bonds between two cysteines. So the cysteines may be far away from one another in the primary amino acid sequence. They could be close to one another. in the primary amino acid sequence, what governs, what forms a disulfide bond versus the two cysteines remaining alone by themselves, just being reduced. We don't know, but the protein knows what it needs to do to fold into its proper three-dimensional structure to perform proper activities for the cell. All right. And the final one is quaternary structure. And I'm not going to go into too much details about quaternary structure, but the classic example is hemoglobin. And a definition of quaternary structure is protein domains interacting with other protein domains. Or proteins, I will just avoid the term domains for now and just say protein-protein interactions. All right, classic example is hemoglobin. Hemoglobin actually has 4 subunits. So this is an independent protein unit that has alpha helices, beta sheets, and random coils. This is an independent protein unit that has alpha helices, beta sheets, and random coils, though hemoglobin is fairly an alpha helical rich protein, it's worth mentioning. This is its own independent unit, this is its own independent subunit, and they join together to form a tetramer. A lot of proteins in cells will form multimers, tetramers, dimers, trimers. It sort of expedites the reaction the cell or that protein is going to perform. So there's some sort of cellular advantage by having proteins not just be one independent entity, but to have several combine themselves into, in this case, like a tetramer, for example. That sort of expedites its main function. In this case, hemoglobin's function is to transport oxygen, and so by it being a tetramer, you actually get more oxygen binding than it being a sole monomer. So this subunit, we'll call it alpha, we'll call this one beta, we'll call this one alpha, we'll just call this one beta, we'll call this the alpha subunit, beta subunit, we'll just call this beta 1, alpha 1, that's just arbitrary for now. But the alpha subunit, this independent folded protein, interacts with this independent beta subunit, that protein, usually through non-covalent interactions. Okay, non-covalent interactions. This alpha 1 interacts with beta 1 and beta subunits through non-covalent interactions. Again, your polar-polar side chains, non-polar, hydrophobic. Okay. These are driving forces. It's important to realize that these drive a protein to go from a linear chain of amino acids to a very beautiful, complicated, and precisely folded native three-dimensional structure. And if it does not fold into its proper three-dimensional structure, you can have a lot of problems. Okay, so that is a quick review of all 4 levels of protein structure. Consult the PowerPoint, use your notes, use the notes that I've printed out for you for a more detailed explanation and for more thorough analysis. But for right now, this video just gives you the very surface level basics of what a primary structure is, secondary, tertiary, and quaternary structure. If I were to say by experience, many students fail to understand what secondary structure really means. Okay, you get an alpha helix, you get a beta sheet through bonding of the peptide bond. Okay, you get these formations, these secondary structure formations, through hydrogen bonding and hydrogen bonding alone from one peptide bond to other peptide bonds around the polypeptide backbone. Okay, so primary, secondary, tertiary, and quaternary structure, know what these mean. We are going from a very simple primary structure, linear chain of amino acids, from secondary, tertiary, and then ultimately to quaternary. We increase in complexity, we increase in three-dimensional folding, we increase in compactness and order of the protein, and all of this is very important for that protein to convey its very specific function within the cell.